Our next speaker is Howard Chang, who is an MD, PhD, and a professor of dermatology, and the director of the Center for Personal Dynamic Regulomics at the Stanford School of Medicine. Howard is also an investigator of the Howard Hughes Medical Institute. His research has illustrated the important and pervasive roles of long non-coding RNAs in biological regulation. He's interested in large-scale transcriptional programs as defined by RNA effectors, including RNAs that mediate epigenetic changes to chromatin. And he's also interested in RNA structure and how it influences RNA function. The title of Howard's talk today is Genome Regulation by Long Non-Coding RNAs.
Good morning. Can you hear me? Yes? Okay. Thank you, Lynn, for the kind introduction, and thank you to the organizers for the opportunity to share our work. So I'm going to talk to you today about how large sets of genes work together, particularly with the help of long non-coding RNAs. So the new century brought us a major biological mystery. As Lynn and Phil pointed out, the completion of the human genome led quickly to the recognition that the vast majority of the genome is transcribed like this, producing many kinds of long non-coding RNAs, at least over 10,000 of these are stably accumulating. But unlike messenger RNAs, whose domains and functions could be inferred by simply checking the sequence database, when we try to do the same with non-coding RNAs, we get a big question mark. And that immediately raised a number of questions about what might be potential functions of long non-coding RNAs. Could they be involved in human diseases? And what might be a way to systematically decode the information content in these new kinds of genes that I'm going to be pronouncing as LncRNAs. Okay. And so in thinking about this problem, I want to take you back to a historical context, because this is somewhat like decoding a lost language. These symbols I'm showing you are Egyptian hieroglyphics, and the meaning of which were lost for hundreds of years, until Napoleon's soldiers found this tablet, the Rosetta Stone, that had the same inscription written 3 times, once in hieroglyphics, once in Egyptian, and once in Greek. And by systematically comparing the text between these 3 languages, they were able to work out the correspondence between letters that we recognize and these ancient symbols. So for example, this text here highlighted in yellow, it's actually a name, Ptolemy. Okay, so it's written more than once, and they could work out that, for example, this symbol must be the same as that symbol in a different language. Okay, so once this correspondence was discovered, they were able to not read only this tablet, but really all the language, all the inscriptions on ancient monuments written in hieroglyphics. And so I think this lays out the task before us. What we need to do is to systematically understand the form and function of these RNAs and then relate them to each other. And so in my talk, you'll hear this kind of logic of trying to, and this is our approaches to understand LncRNA function, to discover novel RNAs in the context of disease and also development, to describe their functions, especially in terms of information transfer inside of cells at a molecular level and in vivo, but then importantly to understand systematically the shapes of these RNAs and how they relate to the function, and hopefully to decode that information. And a lot of that task that involves actually making new RNA tools, because at the time, the methods to actually describe these RNA functions did not exist. Okay. So one of the first examples that we came across was the recognition that RNAs could act as potential guides. So one of the emergent themes for nuclear non-coding RNAs is that they can act as a guide for chromatin remodeling complexes. And that is because many of these enzymes that act on chromatin lack intrinsic sequence specificity and hence need a guidance mechanism. At the time that we started, only a small number of LncRNAs were known, such as Exist and the one involved in dosage compensation, and they all acted in cis, that is, on neighboring genes. So this RNA will be transcribed, recruit a chromatin remodeling complex and enzyme, it will place a histone modification, which actually in this case leads to gene silencing. The RNA we characterized was named Hotair, and this turned out to be the first example of an RNA that acts at a distance. Hotair is made from the human HOX-C locus. It recruits actually one, in fact more, chromatin remodeling enzymes to hundreds of sites across the genome, thereby affecting gene expression, causing gene silencing. The physiologic function of Hotair is to basically program the positional identity of cells. So Hotair was first discovered in human cells in terms of programming skin positional identity. When we knocked out the mouse Hotair, we can see in the skeletal system multiple defects that indicate that segment identity had been transformed. So if you look at each of these bones in the spine, they have a unique distinguishing feature. So for example, this lumbar 6 segment has these lateral processes. The one bone below, the sacral segment, has the iliac crest that's joined here. In the Hotair knockout, the L6 bone is now basically turned into an S1. You see the lateral processes are here, but now the S1, the iliac crest is now joined one segment above. The reason this happens is that the normal complement of Hox genes, these are homeodomain transcription factors that define segment identity, their expression patterns now change. So where in this knockout, these extra Hox D genes are now expressed ectopically, where they should, beyond where they should be. And that's why the more posterior segments take on the identity of a more anterior one. Okay. And so this process is actually not only important in development, turns out to be critically important in cancer. And that is because cancer cells can come in via this process of positional identity. They can reprogram themselves to think they belong elsewhere in the body and therefore allow them to spread to metastasize. Several years ago, we found that, in this case, human breast cancers, about a quarter of these early cancer samples express very high levels of Hotair. When I say high, the cutoff is 250-fold or more beyond normal. When this happens, these patients have a very high risk of eventually developing metastasis and death from their disease shown here. So Hotair is not only a powerful biomarker, it's also actually a driver of disease. When we program human cancer cells to express Hotair at the level seen as that seen in patients, we saw that they were able to actually now basically very effectively colonize the lung, metastasize the lung, and also spread to other parts of the body. So actually a driver of metastasis. So since that time, high levels of Hotair has been shown to be a poor prognosis for some of the most common human cancers. And now many LncRNAs have been found to have these kind of roles as biomarkers. In 2013, the FDA approved the first LncRNA diagnostics for cancer, so it's a very active area of investigation. Okay. In terms of molecular mechanisms, we found that the LncRNAs could actually have multiple roles. In addition to serving as a guide, they can act as a scaffold, as Phil began to describe to you. Early on in development, the histone marks that we're talking about specifying activation or development are actually both present. And this is called a poised state where both kinds of histone marks are present. We discovered that Hotair, the reason why it's long is because it actually has multiple regions or multiple domains. There's one portion of the RNA that basically recruits one chromatin remodeling enzyme that places a mark for silencing. There's a second part of the RNA that recruits a second enzyme complex, the LSD1 complex, that actually removes this activating mark. So the dual action of these two complexes then enforce a clear message to silence that gene. So this example highlights the idea that LncRNAs can be modular. They can enforce proximity and targeting. So this concept, I think, has been acted on by evolution to really create a surprising diversity of LncRNA mechanisms. I'm just going to show you some examples that we came across. So LncRNA can act as guides. There has both a component that the RNA can target a region in the genome and chromatin, and a second portion that can recruit a protein complex. So this mechanism can either activate or silence genes. The RNA is involved in selecting the gene to be targeted. The protein cargo determines the consequence of activity, activation, or silencing. And this kind of concept is also now used in design of synthetic, for example, CRISPR RNAs for CRISPR activation or interference using the same design principle. In the second way, the RNA can also be used as a scaffold, as Phil mentioned to you, bringing multiple complexes together into enforced proximity. In other examples, it can act as enhancers, looping, connecting nascent site of transcription to the target gene, again, bringing protein complexes. And finally, when the gene regulation is done, we can deactivate a program with decoys. And these are RNAs that would, for example, bind to a protein complex and then remove them away. And this requires really a high molarity, a sufficient number of copies to enact these goals. Okay. So this kind of modular design seems very exciting, but it's clear that we're working through these examples in an anecdotal fashion. So I want to then talk to you about really a set of projects we've been working on, I think for actually now almost 5 years, to try to achieve a kind of a predictive or systematic understanding of LncRNAs. And then to do that, I have to introduce a classic genetic and epigenetic system to you, and that is in the Drosophila dosage compensation system. So in flies, the males and females, of course, differ from having males having only a single X chromosome and a Y, and females having 2 X chromosomes. And so the way compensation is achieved is that single X chromosome in males, the gene expression activity is boosted up twofold, again by the action of LncRNAs. And there are 2 LncRNAs called ROX1 and ROX2. These RNAs coat the X chromosome. They recruit a set of chromatin modification complexes, the names of which are not important for now. But these RNAs are required to do that. And then in the absence of these RNAs, so if you knock out either one of them, the organism is okay. But if you knock out both, all of the males are 100% dead. All the females are absolutely fine. So this is a very useful property we can use in our genetic studies. The other thing I want to point out to you is that these 2 RNAs, despite being genetically redundant, look nothing like each other. ROX1 is 600 nucleotides, ROX2 almost 4K, sorry, the other way around, ROX1, 4 KB, ROX2, 600 nucleotides. If you try to search for them from each other using BLAST, they don't even come up as the top one in the top 100 hits. So this just shows you the challenge, the difficulty of recognizing LncRNA orthologs or functional homologs. Okay. So we started working on this, trying to understand at high resolution the mechanism of RNA targeting to chromatin. And we developed a technology called chromatin isolation by RNA purification, or ChiRP. So just like CHIP technology that Phil describes to you, this is a technology where we take cells, the endogenous RNA, we cross-link them to trap the endogenous interactions. Then we come in with oligonucleotides against an RNA of interest, so we can retrieve the endogenous complex of DNA and proteins, and then we can isolate them and then go on to deep sequencing or other detection technologies. So when we apply this technology to the ROC system, we improve the ability to map the location of this RNA from a megabase scale, from traditional fish experiments, to basically, I think, near dozens of nucleotide sort of resolution. So we can see that across the x-axis, this is genomic coordinates, we can map in very high resolution the locations of these ROX2 binding sites. And they actually line up precisely with genetically defined chromosome entry sites. These are high affinity binding sites for the dosage compensation complex. We can also map that across basically all genes across, let's say, the fly genome, and show that it's only on the X chromosome, not on autosomes, and actually it's over the gene body, extended actually more and more towards the 3' end shown here. So then, taking this kind of concept, we realize we can actually take this one step further to dissect RNA modules. This is a strategy now we call domain ChiRP. So instead of deploying your affinity reagents, let's say all at once, we now design sets of reagents that target, let's say, the first 100 nucleotides, the second 100, the third 100, and walk your way down the RNA. It's an RNA-centric method, and when we retrieve each sort of pool, segment of RNA, we ask, are we pulling down other parts of the RNA? Are we pulling down other proteins? And are we pulling down DNA? So in this way, we can dissect the pairwise interaction of that RNA to every other component. And we can do all this in situ without doing a lot of cloning or making deletion mutants. Okay. So I'm going to highlight what we learned from this process going to the increasing resolution. So when applied to ROX1, one of these key RNAs in the Drosophila dosage compensation complex, we realized that the architecture of the RNA looks like a 3-finger hand, okay? So these fingers, which we're calling D1, D2, and D3, they each pull down only themselves and the other parts of the RNA. Whereas the central core, if you pull down U1, you will pull down U2 and U3, and vice versa. So that's one difference. Second point is that these fingers are parts that are binding to both chromatin and the chromatin remodeling complexes. All the proteins and the DNA interaction come with these fingers. Now I'm going to show you in high resolution what these fingers actually look like. Okay? This required really developing some other methods to map RNA structure, both in living cells and in vitro, again, at a genome-wide, transcriptome-wide scale. And so I'll just show you that this is a method called SHAPE that measures the single-stranded regions of RNA. This method, PARS, measures both single and double-stranded regions. So we can work out that one of these regions is actually 2 stem loops, a tandem stem loop just like this. I want to point out to you that you notice that if you look at the conservation map, only the stems are conserved, okay? And then the loops are not, and the spacer sequence is not conserved. It turns out that the spacer is exactly, usually, 22 nucleotides. It could be any sequence, but it just has to be that long, okay? But the stems have to be conserved, and on the 3' arm of the stem has to be a certain motif, and that's exactly where the binding occurs to the key protein complex. This is the eclipse signal showing that's where they bind. So this is the fundamental unit of this long non-coding RNA. This is the structure, and this is how this starts to be recognized by the machinery. Okay. Let me show you some proof that this is actually true. And so one of the key recognition of this technology was that we recognize that when we look at this long non-coding RNA, only certain parts of the RNA actually matter, and we also know which ones matter the most. And so, for example, in this long ROX1 RNA, this D3 finger, we think, contained all the information. And so the experiment we did was to basically take a ROX null male fly, which would all die otherwise, rescue with a genetic transgene, always knocked into the same position and with powered Drosophila genetics, and ask, can you rescue? And it turns out this D3 finger can rescue as well as the full-length transgene. So essentially, we've reduced the size of this long non-coding RNA by 10-fold to a mini gene that suffices for chromosome-wide dosage compensation. Okay. So we want to now take this one step further. If we have this kind of understanding of a single LncRNA, can we look across now new genomes, new species, to identify LncRNA orthologs? The strategy we took is something that we're calling nested homology, because we use multiple forms of information to find these otherwise invisible LncRNA genes. So we first look at whether the genes are sharing gene neighbors, or synteny, and this is important for regulation. This is an idea developed by David Bartel, which we incorporate. Once we have a candidate region, we search for these sequences that might be repeated, because our observation was that these LncRNA domains are often repeated multiple times. That's what we call that microhomology. Then we further search these individual sequences to see if they match up to these RNA structural sort of key elements that are present. And if the answer is yes, that basically counts as a hit. We then go on to the next species using the one we just found to bootstrap as the kind of basis, as the seed for the next search. Okay. I should point out to you that this technology, this strategy was successful, but the amazing thing is that these LncRNA orthologs have very low primary sequence identity. They're not really any different than perhaps background. So if you're not looking for these key features, right, you would not actually recognize them as actually being orthologs. Okay. So to show you how this works, we apply this to across the Drosophila evolution, going back 40 million years through these many, many different Drosophila species. And so just to put things in context, 40 million years of divergence is like looking at different kinds of mammals. It's like comparing raccoons to brown bears. They're really quite different. And to just make that point, even the identity of the X chromosome is actually starting to be different across evolution. Drosophila have 6 chromosome arms. They're called Mueller elements, A through F. And so in melanogaster, the Mueller element A is the X chromosome. But in some of these other species, now we have a Mueller element D or C or so forth that are now the X chromosome inherited in a single copy in the male, needing dosage compensation for viability. Okay. And so all we have here is the draft genome sequence, sometimes not even the whole genome sequence, just the DNA sequence. And using the approach that I described to you, we're able to identify 46 new ROX orthologs, essentially now tripling the number of ROX RNAs that have been discovered. I'll show you evidence whether they're actually true orthologs and functional. Okay. I want to summarize some key points. So first is that the LncRNAs are rapidly evolving. Okay? So despite the fact this is an absolutely essential process, and I think all the flies need to do dosage compensation, there are a lot of changes to the LncRNAs. Sometimes there are actually 3 ROX RNAs in the sultans, Willistonea subclade. There are actually 3 ROX RNAs. I will show you that in some species, the ROX1 has completely degenerated. The entire job is being done by ROX2. And there's some very interesting sort of division of labor. So if we have the presence of synteny, that is sufficient to confer regulation. So all the ROX orthologs that we found are expressed in a male-specific fashion. But despite the fact the RNAs have this interesting expression pattern, that does not mean that they're actually sufficient for function. Many of them, actually, as I told you, the function has changed, and that understanding absolutely requires an understanding of the RNA structure. If you have the key elements, you have function. If you don't, you can be expressed, you're not going to do anything. Okay. So I'm going to focus on studying 4 species that are basically, we've chosen them because they're perfectly spaced across this evolutionary distance: melanogaster, Willistonea, virilis, and buskii, which I'm going to name in the subsequent talks D-mel, D-will, D-vir, and D-bus. I'll always show you those species names on the side. So one of the approaches we took to understand the evolution of LncRNA genome interaction is to perform ChiRP across these 4 different species. So we developed a way to do this in vivo from the larvae of these flies. These are actually not actually laboratory organisms. So it's actually, I have to give credit to my lab members for actually learning how to grow and sex all these different kind of flies. Okay, so looking at the ChiRP signal for ROX1 RNA or ROX2 RNA across these 6 Mueller elements, you can see that, as expected, in Drosophila, it's only on the X chromosome Mueller element A for both ROX1 and ROX2. And in these other species, the ROX2 is always on the X chromosome. So for example, in Willistonea, both A and D elements are the X, and so it's both on A and D, but not other chromosomes. So there already is a very interesting evolution of the expansion of the binding site to the D Mueller element. But you see that there's much less signal for the ROX RNA. This is quantified here. Okay, so the ratio between ROX1 versus ROX2 binding signal to the X chromosome is nearly 1 in melanogaster, really bad in buskii, and getting progressively better. This can be systematically understood by looking at the sequence and the structural level of these RNAs. So ROX1 in Drosophila melanogaster has the complement of these tandem stem loops. This is now missing, D-will is missing some, and it's getting even worse, missing 1 more. And in D. buskii, there's even a competing secondary structure to this preferred structure, making this an even worse candidate. To prove that these kinds of inferences are actually true, we then come back and try to do genetic rescues of these other species, ROX RNAs, to rescue the ROX knockout melanogaster. Okay, so again, these are null flies that would otherwise all die. This is the D. virilis ROX1, the D3 finger. You can see that actually has substantial rescue of this, the Drosophila knockout. Okay? Now, furthermore, we can show that it really is the number of these repeats that matter. If we add 1 more from melanogaster right here, we now get substantially better rescue. Finally, the next experiment really surprised us. To understand how important are these individual stem loops, we made a synthetic RNA that comprises basically a single stem loop, but fused to the LAC-Z, bacterial LAC-Z sequence, which is an artificial LncRNA. And even this RNA actually can rescue almost 2%. Okay? So you might think that's not very impressive. I don't need to tell you that this system is actually extremely stringent. in the roc's null background, basically we get less than, we screen over 10,000 flies. You don't get a single viable male fly. So the background rate is extremely low. So this 2% rescue is a 100-fold improvement in viability for this genetic background. So we think that if this were to occur in evolution, there would be a very strong driving force for this to be preserved and then perhaps expanded. Okay. All right. So we can now look on the other side of the question, how does the genome accommodate the changes in LncRNA evolution and sequences? And so this is now mapping the top motif on the DNA side that comes out from rock's occupancy. Again, D. mal, D. will, virilis, buskii. The top sequence that emerges is the same in all these species. There's always this GA, GA, GA motif. This is exactly the same. That may imply that, wow, this is a very conserved system. These elements should really be staying put across evolution. But when you zoom in onto the genome, you see something different. So we do, in fact, have some cases where the location of the binding sites is exactly the same across these 4 different species. So it's shown in order. But that's a minority. Most of the time, we see that these binding sites keep moving around. Here, 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 in a surprising fashion. So that is the predominant pattern. I'm now going to summarize this observation for you across all sites at the level of genes. So these are the 4 species we're looking at. Now I'm asking whether if I see a binding site in one species, is that gene also occupied by rocks in another species? So you can see that these are all the sites that are bound in all 4 species, all 4-way invariant. That's very few. In fact, the most frequent occurrence are these single species specific events, right? Only bound in one species, but not in the other. So how can we explain that? This seems like a conundrum. So when we then zoom in to look at the binding site in one species and ask, what is the next binding site in the ortholog species? We see that they tend to occur much closer to each other than expected by chance alone. And that is, if you have a binding site in one species, you may not have another site in the other species, but you have another one very close by. Okay? And so these binding sites are acting like cell phone towers. You don't have to have them exactly at that position, but you have to have them in the neighborhood. And so we can tolerate essentially. Let me just explain to you what's going on here. So this is mapping the distance, the frequency of getting a distance, another binding site within a certain distance constraint. This is what is observed. This is what's expected by chance alone. And we see essentially that you have a much higher chance of getting another site within 25 kb to 50 kb range for these binding sites. So what this observation is telling us is that these individual elements are disappearing, but replaced by new elements nearby. But for this to be possible, we now have another conundrum, and that is that these new elements must be appearing quite frequently to make it possible for the old ones to go away. So how are the new elements appearing? We have a very good system to look at that question in this data set, because we can look at these single species specific elements and ask, what is their identity in these other species, which these elements are not yet a ROX binding site? What are they? Okay. So here we have I think a very satisfying result, and that is how we discover that the DNA sequence encoding an RNA splice signal moonlights as future ROX binding sites. So I'm plotting for you the location across introns, and I'm showing you that the density of these ROX binding sites, they're called high affinity sites, HAS sites, there's a huge peak precisely at the position of a signal called the polypyrimidine track. This is a signal needed for splicing. It is a CT or CU-rich sequence. It could be CU-CU or CCUU. What we recognize is that the reverse complement of this sequence is very close to this GA-GA-GA sequence. And so the assumption is that if you have this CT-rich sequence, by simply shuffling the order a little bit, you can preserve the functionality as a polypyrimidine track, as an RNA splice signal, but at the same time gain a functionality on the DNA side as a ROX binding site. The diagnostic test for that concept is asking for strand bias in the occurrence of the polypyrimidine tracks, because this model predicts that always basically the ROX binding sites in introns should show this very strong strand bias, where the forward strand is basically the CT repeat, the reverse strand is a GA repeat, and that is exactly what is observed here across the X chromosome with a very significant value, indicating this is unlikely to happen by chance alone. The reason this observation to me was satisfying is that it actually couples dosage compensation to splicing. And this solves actually very elegantly a problem that evolution encounters. As I mentioned to you, in the Drosophila evolution, different chromosomes can actually get joined on as the X chromosome. All of a sudden, you have a new chromosome that you have to compensate for. How do we do that? This mechanism ensures that we target the dosage compensation machinery only to the actively transcribed and spliced genes. You don't waste your energy on sequences that are never transcribed or spliced. So I think it's a very elegant solution to this potential problem. Okay, I've told you now that I think the surprising result is that despite the fact that this is an essential and conserved mechanism, always the X chromosome, always the same motif, it's actually undergirded by very rapid evolution, both on the RNA and on the genome side. On the RNA side, I told you that there are these individual elements that are refined by repetition to gain activity. And on the genome side, I mentioned to you that there are this fast turnover in proximity, where basically by reusing existing sequences, we can actually repurpose them for a different function to gain, for example, a binding for a specific RNA sequence. In fact, basically gaining and repurposing LncRNA genome interactions. Okay. So in summary, I've told you about really the interplay between RNA and chromatin. LncRNAs are a major force, I think, in changing chromatin biology and driving changes chromatin activity. And I'd like to posit now, because of the flexibility and adaptability of LncRNAs in evolution, they may act as innovators of gene regulation across evolution, as I've shown you, to preserve and create key biological activities and organismal possibilities. I'd like to thank many lab members that have contributed to this work. The work on Hotair was pioneered by John Rinn when he was a postdoc in my lab. Fortunately, I'm happy to report that he's now a full professor at Harvard Medical School. Other members who contributed important aspect of the work, C. Chu did the work on ChiRP development. The work on ROX evolution was all by Jeff Quinn, who has normally, this is I think a Halloween kind of a presentation, and with important collaborative work, support from Asifa Akhtar's group that did a lot of work on the Drosophila genetics. And I'd like to thank my funding sources. Thank you for your attention.
We have 2 questions before we have a break. So, Jeannie and then.
So one of the key properties of these ROX RNAs is their ability to coat the X chromosome. So I'm wondering whether these new ROXs that you're discovering in parallel species, whether they also coat the chromosome, and do you know if they recruit the MSL complex in the same way?
Yes, the answer is that they coat the chromosome. So if you do in addition to the ChiRP, if you do the fish experiment, they coat the chromosome. They don't actually always use the MSL complex though. That's the interesting thing. We have some evidence that they actually could use some different mechanisms, some of these other species. Yeah.
It's a very nice talk here.
Thank you. Oh, here. Yes.
So my question is, it seems that the secondary structure of the LncRNA is very important to its function. So my question is to what extent the prediction method, for example, the RNA fold prediction agrees with your shape experiment data?
Right. So yes, in this particular case, the RNA fold predictions and also the evolutionary covariation supports that. But I think that we're of the view that it's important to have experimental data guiding the RNA structure prediction. The predictions will be much better guided by actual experimental data. And that's why we put a lot of effort into using different methods, shape, parse, and other methods to actually experimentally map the RNA structure, which is not possible in living cells and also transcriptome-wide. We think that is a key aspect to understanding RNA function.